In this chapter, we'll be looking at the elements of chemotherapy, or treatment with drugs. Now, it wasn't more than 100 years ago that 1 out of 3 children were expected to die before the age of 5 from some sort of infectious disease, and others would be permanently maimed by those diseases. It wasn't until the 1930s that we saw our first drugs for the treatment of infections. These, of course, were regarded as miracle drugs. But even still, we've not eradicated infectious disease. In fact, on our present path with the overuse of antimicrobial products and antibiotics, some suggest we have the potential to get very close to a post-antibiotic era where our drugs are no longer effective against the resistant strains of microbes that have come about. Now, it's as recent as 1941 that penicillin was in its clinical trials. So we've come a long way since then, but we need to be really careful. So let's begin by taking a look at some of the principles of antimicrobial therapy. The goal of antimicrobial therapy is to administer a drug to an infected person, which destroys the infective agent without harming the host cells. Well, that seems like a relatively simple concept. However, it's pretty difficult to achieve. Many, often contradictory factors need to be taken into account. And microbes and biofilms often require different drugs than when they're living independently. Over on the right here, there's a figure that summarizes what an ideal drug should be. First, it should be selectively toxic to the microbes, but not toxic to the host cells. We would like it to be microbicidal rather than microbistatic. Relatively soluble, so that it functions even when it's highly diluted in body fluids. The ideal drug should remain potent long enough to act and not become broken down or excreted prematurely. We can also be hopeful that it doesn't lead to the development of an antimicrobial resistance, as many of our drugs do. The drug should complement or assist the activity of the host's defenses, remain active in tissues and body fluids, be readily delivered to the site of infection, be reasonably priced, and not disrupt the host's health by causing allergies or predisposing the host to other infections. If you've ever listened to a commercial on television about side effects of drugs, you'll recognize that it's virtually impossible to meet all of these characteristics of the ideal antimicrobial drug. We will be covering chemotherapeutic agents with regard to their origin, their range of effectiveness, and whether they are naturally produced or chemically synthesized. There are some important terminologies of chemotherapy in the table to the right that you should take a quick look at before we move on to the next section. In the following slides, we'll cover different types of antibiotic drugs. We'll look at their mechanisms of action and the types of microbes on which they are effective. Now, most antimicrobial drugs that we have in our arsenal today have been provided by nature or modeled after those that are provided by nature. Most antibiotics are common metabolic products of aerobic bacteria and fungi, and the greatest numbers of them have been derived from a couple of species of bacteria and a couple of molds. Specifically for bacteria, it's been the Streptomyces genus and the Bacillus genus, and for molds, Penicillium and Cephalosporium. Chemists have created new drugs by altering the structure of these naturally occurring antibiotics. Now, why might microbes produce their own antibiotics? Well, the answer probably lies in the fact that they use these antibiotics in order to inhibit growths of other microbes in their own environment, a form of antagonism so that they can survive preferentially over their competitors. Presently, we're searching for metabolic compounds with antimicrobial effects in species other than bacteria and fungi. Fairly recently, we've discovered that Hydra, which are microorganisms that are well known for regenerating parts that have broken off. Well, it's been recently established that they produce a compound that's now been named Hydramacin. This is a protein, and it's active against both gram-positive and gram-negative bacteria. Now, usually antibiotics aren't protein in structure. Most of them are very small molecules that contain some ring structures. So this is some pretty exciting new material. Now let's take a look at how drugs affect microbes. The goal of antimicrobial drugs is to disrupt the cell processes or structures of bacteria, fungi, and protozoa, or inhibit virus replication. Most drugs interfere with the function of enzymes that are required to synthesize or assemble macromolecules, or they destroy the structures that are already formed in the cell. Drugs need to be selectively toxic. That is, they need to kill or inhibit microbial cells without damaging the host cell tissues. There are a number of different microbial targets. First, we can inhibit cell wall synthesis. We could inhibit the nucleic acid structure and function. We could inhibit protein synthesis, or interfere with cell membrane structure and function, or inhibition of folic acid synthesis. This figure summarizes all of these different approaches, and also gives you some idea of which antimicrobials have what effects. This is a great summary figure for your studies. Let's first look at antimicrobial drugs that affect the bacterial cell wall. Most bacteria have rigid peptidoglycan cell walls. They could either be thick and gram-positive or thin and gram-negative. And active cells have to constantly synthesize new peptidoglycan and transport it to the proper place in the cell envelope. Drugs like penicillin and cephalosporins react with one or more of the enzymes that are required to complete this process. For example, they can block peptidases that form the peptide cross bridges that you'll recall are evident in peptidoglycan cell walls. So if there are no cross bridges, then weak points develop in the peptidoglycan cell wall, so that the membrane then bulges out between these weak points and eventually ends up lysing the cell, because there's no longer a cell wall there to prevent cell explosion as water diffuses into the cell. These antibiotics can only be active in young and growing cells because those cells are making peptidoglycan constantly. Cells that are old or inactive are no longer producing peptidoglycan, so this sort of antimicrobial drug is ineffective in that case. This class of antibiotics are considered to be bactericidal antibiotics because they actually cause complete destruction of the cell through cell lysis. Here are some of the other drugs that target the cell wall. Bacitracin is from the Bacillus subtilis and is found in Neosporin. Isoniazid is usually used for tuberculosis. Vancomycin is used when people have penicillin allergies or in MRSA attacks. Now we'll take a look at antimicrobial drugs that affect nucleic acid synthesis. They can either block synthesis of the nucleotides, inhibit replication, or stop transcription. Because DNA and RNA are required for transcription as well, the effects on protein metabolism can also be fairly far-reaching. We'll take a look at those shortly. Other mechanisms some of the drugs exhibit are cross-linking of the DNA double helix, like we see in chloroquine, which is an anti-malarial drug. Or we can see inhibition of helicases, so the DNA cannot unwind. This is seen in quinolones. Also in antivirals, like azithromycin, AZT, or acyclovir, we see that they form analogs of purines and pyrimidines and insert themselves into the viral DNA so that DNA replication is halted. As we've already seen, many antimicrobial drugs target protein synthesis. Again, in the process of transcription, it's a fairly difficult target because the process is pretty well conserved. However, rifampin is a drug that binds to RNA polymerase and prevents elongation of the transcript after initiation. It's specific to bacteria, and it's often used in treatment of tuberculosis. Now, translation is the place in protein synthesis where the majority of antibiotics are targeted, simply because the ribosomes of prokaryotes are different than eukaryotic ribosomes. There are 70S ribosomes. They have a 30S subunit and a 50S subunit. So the small subunit and then the larger subunit. Various antibiotics inhibit translation by reacting with that ribosome and messenger RNA complex. Here are a few examples. Some antibiotics bind to the 30S subunit, and they'll cause misread in the messenger RNA so that the protein that's produced is incorrect. Others might bind to the larger subunit and prevent formation of peptide bonds, whereas others might actually bind to one of the active sites on the subunit and prevent initiation and thus block the entire ribosome assembly. And others, like the tetracyclines, tRNA is blocked, so no protein synthesis can happen because it can't enter the A site on the ribosome. Erythromycin works by preventing translocation of the messenger RNA into the various active sites of the ribosome. So there's a number of different ways that antibiotics can target the ribosome during translation. Again, you'll find this figure as an excellent summary. Let's now go on to take a quick look at how antimicrobials can be involved in the prevention of protein synthesis.
Several types of antibiotics inhibit bacterial protein synthesis. The 70S bacterial ribosome, which is composed of 30S and 50S subunits, is sufficiently different from the 80S ribosomes of eukaryotes to make it a target for selective toxicity. Most antibiotics bind either to the 30S or the 50S subunit of the ribosome, thereby interfering with protein synthesis. Aminoglycosides such as streptomycin bind to a protein in the 30S ribosomal subunit and interfere by inhibiting protein synthesis and causing misreading of the mRNA. Different aminoglycosides bind to different target proteins on the 30S ribosomal subunit. These antibiotics distort the ribosome so that misreading of the code in mRNA occurs and protein synthesis cannot continue. Bacteria can become resistant to aminoglycosides by mutation of the target ribosomal protein. The tetracyclines inhibit protein synthesis by binding to the 30S ribosomal subunit and inhibiting the binding of aminoacyl tRNA to the A site. In contrast to streptomycin, they do not stop the ribosome on the mRNA, but distort the ribosome so that the incoming aminoacyl tRNA can no longer bind properly to the A site. Bacteria become resistant to tetracyclines by developing an efflux mechanism that removes the tetracyclines from the cell. Some utilize a protein that binds to the ribosome and prevents binding of tetracyclines, and some strains of Bacteroides become resistant by chemically modifying the tetracycline so that it does not interfere with protein synthesis. Erythromycin, a macrolide antibiotic, binds to the 23S RNA found in the 50S ribosomal subunit and inhibits peptide chain elongation. This binding results in release of the peptidyl tRNA. Bacteria become resistant by producing enzymes that alter the erythromycin so that it can no longer interfere with protein synthesis.
Another set of antimicrobial drugs will disrupt the cell membrane function. Damaged membranes invariably result in death from disruption in metabolism or lysis of the cell entirely. The specificity for a particular microbial group is based on differences in the types of lipids in their cell membranes. As we've seen previously, detergents will bind to the phospholipids and also lipid A in the cell membrane. These can disrupt membranes through their surfactant action. However, there's poor selective toxicity because all cells have phospholipids in their cell membranes, and so there's a high risk of destruction of the human cells too. Polymyxins are found in triple antibiotic creams. Of course, these have to be applied topically. Again, they interact with the membrane phospholipids. Finally, we'll take a look at a group of antimicrobial drugs that inhibit folic acid synthesis. These are the sulfonamides, or sulfa-based drugs. Sulfonamides and trimethoprim are drugs that act through competitive inhibition, so they get in the way of metabolic pathways, particularly in this case, metabolic pathways that are involved in the production of folic acid. So you can see in this pathway that PABA is converted eventually to folic acid. There are several enzymatic steps in the pathway, and sulfonamides inhibit the enzyme that convert PABA into the next intermediate in the pathway. If this is broken, then we never get the formation of folic acid, and thus purines, pyrimidines, and amino acids cannot be made. Often sulfonamides and trimethoprim are used in conjunction because they have a synergistic effect. That is, both drugs given together have more than an additive effect, and it requires lower doses of each drug for the desired effect. So essentially, when they're prescribed together, they work better with lower doses. And it basically comes down to the enzyme is supposed to pick up PABA, but sulfonamide looks very similar to PABA in its docking region, and so it's absolutely capable of going into the active site. The more sulfonamides that are present, the more of them bind into the active site, and thus the more inhibition there is for the production of folic acid. And if folic acid is absolutely necessary for the production of these guys, then the cell is unable to metabolize and grow. So sulfonamides and trimethoprim are selectively toxic only because mammals get their folic acid in the diet, whereas microbes have to metabolize PABA in order to produce folic acid. Before we move on, let's have a quick concept check. Which molecule do sulfonamides disrupt formation of? Peptidoglycan, glycogen, or folic acid? This one's pretty straightforward because we just finished it. Folic acid production is prevented by sulfonamides. But what gets in the way of peptidoglycan formation? Ah, right, penicillin.